So thank you, Maya, for the introduction. So my lab is focused on tilapia. Tilapia is the most cultured species in Israel and the third most cultured species on the globe. And we're trying to understand how things work within the fish and to utilize that to improve it by genome editing. So it's not a secret. There's an explosion in human population, and it continues to grow, and there's not enough food, especially not animal protein, and more than that, not enough aquatic animal protein. You can see in the red and orange, in the last 30, 35 years, there have been no increase in the success of fishing in wild regions, although technology, everyone knows, that significantly improved. And this is because the oceans can't suffice. They're becoming empty, and the only solution is to perform aquaculture, which you can see in the light and dark blue. And this is continuously growing. I didn't think of that, yeah. Okay, so this is why aquaculture is important, and this is why this is my passion. So obviously there were fish researchers before me in Volcani. Early works were focused on genetic programs for the coloration of tilapia, improved stress tolerance and sex determination in Nile tilapia, and also mapping of coloration genetics in ornamental carp. You know them probably as koi. But probably the most known work that was performed in the Volcani fishery was the establishment of the DOR 70 strain. That's a strain of common carp, which is also called mirror carp because it lacks a lot of scales. And this is one of its main advantages in addition to improved growth and increased survival rate. And these are the main findings that are somehow related to my work. And when I arrived not many years ago to Volcani, I established my lab trying to understand how things work and gain some mechanistic understanding, but also to try and see the applicative needs of the field and try to join the two. So we're trying to understand how the environment influences muscle growth or homeostatic regulation of response to cold stress, how stress and metabolism are generally regulated in the brain of the fish. And in parallel, we try to perform some advanced applicative research to generate red tilapia phenotypes, increase the growth rate of the fish, increase their appetite so that we can give them cheaper feeds, and to improve their stress resilience. And we try to do that by combining the two within the CRISPR-Cas9 method. This method is basically somewhat of molecular scissors, which allows us to take short fragments outside of the animal's genome, and by that generating loss of function mutations in the animal. And this is a very effective approach to generate reverse genetics in food fish. So what is this method? Basically, it's a small nucleotide sequence and non-specific enzyme. This method gave Emmanuel Charpentier and Jennifer Doudna the Nobel laureate for establishing it. And basically, these two, if we modify them just slightly, we can cause them to target a specific site in the animal's genome, and by that generating double-strand break. And the system can't fix it always appropriately, and this is the basic of the method. By that, we're generating the loss of function indels. So the first thing I tried to do in my lab was to generate red tilapia. Why? First of all, it's very convenient. It's a very easy phenotype to detect. You don't need to work much molecularly to know that it's working. Basically, you lose the pigments on the fish. But more practically, red tilapia has a very high demand and significantly increased market value. both in Israel, but mainly in Southeast Asia, which is a huge market. And additionally, current red tilapia strains are either highly inbred, which suffer from retardation in growth and pathogen resistance, or from hybridization, but then you can see the dark blotches on these fish, and these cause a very significant reduction in the market value of the fish. So trying to establish the method, basically what we did is we calibrated many of these steps that you can see here. Basically, we do in vitro fertilization. We select the appropriate zygotes, because from the moment of fertilization, we have about 20 to 30 minutes to perform the microinjection. We perform the microinjection to the embryos. We hold with one pipette. It's working. Fantastic. So we have a holding pipette, which we hold the egg because it's very hard and tough. And from the other side, we come with a micro pipette and inject straight into the single cell. We transfer the embryos into incubation, and the ones that survive, we transfer to grow out, following which we can do genotyping, check if things worked, and then select our animals for the next breeding cycle. So wild-type embryos at 4 dpf, you can see a lot of melanocytes, hundreds of melanocytes on the embryo, and you can see, I hope you can see, the melanin starting to accumulate within the eyes. And already in the first injections, we could detect that we significantly reduce the melanophores on the skin of the fish. And you can see that, for example, here, oh, hard to. So here, this is the head of the embryo, and because of the lighting, you will need to trust me, but the eyes are here, and they are completely transparent. So at this stage, we knew that this is working, but the next question was, will it go to the next generation? So we crossed the fish, we generated the homozygous mutants, and you can see that there is a significant reduction. Basically, we generated albino fish, and these fish also have red eyes, unlike the strains that currently exist that have dark eyes, which is a hallmark of albinism in many animals. And knowing that we can do this, we thought what would be the next thing to tackle. So obviously, everyone wants more muscle mass in their animals. Why? Muscles are the edible part of the fish and any farm animal. And in addition, more than 50% of the fish weight when it's going to marketing is muscles. So every small advantage in this has a huge advantage for the marketers and for the consumers. And to do that, we chose the pathway of myostatin. Myostatin is probably the most known and most effective inhibitor of muscle growth, and we were thinking that by inhibiting the inhibitor, we can add more muscles. And this pathway is strongly conserved throughout evolution, so this also gave us more confidence that things will go well. One drawback of our approach was that most fish genome contains 2 myostatin genes, while mammals and avians have only one myostatin gene, which introduces some more complexity, but we still thought that this would be the best approach. So this is Belgian blue bull, and you can see that it's highly muscular. Here you can see a wild-type whippet versus a myostatin knockout whippet. This caused an increase of about 100% of muscles. And you can see also here in the red bream, there's an increase. This is a mutant generated in Japan. And you can see that it also has increased muscle mass. And as I told you, most fish have two myostatin genes, but when we started, there was only one gene that was known. So we needed to identify the second one, and indeed we found out that there's one which shares high genomic conservation even up to the human genomic region. The next stage was to try and understand how these two genes express within tissues. This is a good hint many times to understand what's the role of each gene, especially when there's duplications. And you can see that myostatin 1 is strongly expressed in the muscles of the fish. Surprisingly, myostatin 2 is not expressed in the muscles at all. It is mainly expressed in the brain, actually, which goes well with my research interest, but with the theory of regulation of growth. So, as I told you, this is different examples in mammals. Myostatin knockouts increase significantly. These are examples of CRISPR knockouts. And you can see that we can recapitulate the phenotype and increase significantly the muscle mass. But when people did it in fish, and these are just two examples, there's a few others, the increase in the muscle mass is only about 25%. And we still don't know why. One speculation is that because myostatin is an endocrine factor driven from its site of production to the target region, then possibly hypothalamic myostatin is produced, secreted into the bloodstream, and still suppresses some of the muscle proliferation, by that blocking some of the advantage we can gain. Because of that, we decided to do double knockout in tilapia, and indeed we succeeded. We did a multiplex injection. By that, we knocked out two genes in one injection event, and these are just examples of how these things look in the lab. So you can see the wild types in the red, and these are the molecular curves, and you can see the shift in the mutants. And this is an example from a single fish, and nowadays we have a germline of these double mutants, but unfortunately it will take a few more months until we will have the homozygote mutants, so I can't show you any phenotype yet. I had an animation, I'm sorry. I didn't understand what happened. So, surprise. My lab is also doing additional genome editing on several pathways. Some of them, for example, are improved appetite. We still try to see whether there's a good pathway to tackle and generate boneless fish. And I hope that in the Volcani 105, not 200, we'll be able to show you already phenotypes of crossbreeding between all these mutant lines. And then we hope that we will have a red, big, hungry, and possibly with no bones tilapia. And with that, I would like to thank all the great people who did the work, collaborated. Mainly Adi Segev, who is on maternity leave. Thank you, Maya. To Nico, and all the other members in the lab, members from the Volcani fishery, our collaborators from Weizmann and the Yukon, and obviously to the great funding agencies that understood the greatness of our work and financed our games. So thank you all for listening.
Thank you, Kobe. Yes, we're behind schedule, but Kobe did a good job on, so we'll allow a few questions, so go. Just repeat the questions.
Kobe, how does that affect the taste? So the question was how that affects the taste. So there's two parts to my answer. One is we still don't know, but people are eating with their eyes, and red tilapia that are sold nowadays, people say that they are much tastier and pay more because they claim it's tastier, but basically it's more because it looks fancier. Okay? So that's one part. The other part is that Some thoughts are to affect, or we can affect the taste by genome editing, but we are not there yet. So there's two. Sorry. How do we get from the albino tilapia to the red tilapia? So scientifically, there's a debate whether the red tilapia are a consequence of expression of red pigments on the skin of the fish that are seen because of the loss of black pigments, or is it because of the sun reflection on the blood irrigation on a transparent skin? Whatever the case is, I can already tell you that our fish, when they grow, they become a very fancy orange, a very bright and very nice one. It's not completely red, but it's a beautiful fish. I would hold them in an aquarium. And I believe that when they will be out, tilapia are obligatory herbivores. So when they eat algae, this phenotype will intensify even more, but we need to go out to the field to know that. What are the red pigments? So it's all melanin. There's eomelanin and pheomelanin. One of them is the dark red, black and brown, and the other is red and orange. One option we thought of was actually to block the downstream black, because first the cell produces the red pigment and then it converts it to an intermediate form, and then it divides to two clades. One would be synthesis of red and one would be synthesis of black. We thought of blocking only the black pathway, by that actually increasing synthesis of red within black cells. But we went for a simple, more straightforward genetic approach, because the other way would need at least 2 gene knockouts. Thank you very much.
Okay, so thank you all the speakers, Roy, Ellen, and Kobe. Let's thank them again. And we'll be back at 4. Thank you.
So I don't know if it will help to gather more people around here, but okay, so maybe I will start.